Okay, so today we're doing the biology unit 4 paper of June 2015. So this paper is 90 marks. It's over here, and hopefully we're supposed to get an A star, which is anything above 80%. Over here it's starting off relatively simple by showing us the carbon cycle. So we need to complete the diagram by writing the appropriate word or words on the dotted lines. So of course we should know about the carbon cycle. It's how it goes into the atmosphere, into plants, into how it's used by plants, and how it goes back to the atmosphere again. They've already made us a cycle, we're just supposed to fill in. So over here we know that it's going to be carbon dioxide in the atmosphere. I was just gonna write that down like that. Carbon dioxide, just like that. And then over here in plants, it's obviously as an energy store. So I would say glucose, because that's how it's made by photosynthesis. It turns into glucose. And then that's just 2 marks over there. Then we need to explain the role of light in the photosynthesis stage of this cycle. Now we need to explain what does light do. So obviously light is used in photolysis. That's like the first part. Light is used in photolysis. And we need to explain what photolysis is. So photolysis is the breakdown, maybe the splitting. Photolysis is, I'll say splitting. You can say breakdown. I think splitting would be more appropriate here though. So it's the splitting of a water molecule. So it's the splitting of water by photons of light. By photons of light. There it is. That's probably our first mark. We explain how light is used. They're splitting water. What happens to water? We get what? So it's H2O, we know that. So if we're going to split this, we're going to get H plus ions. And the H plus ions are the ones that we need. So I have to explain how. So the broken H plus ions will, what will they do? They reduce. So they reduce NADP to NADPH. So we got hydrogen from there to here in NADPH. And then we have an excited electron. So an excited electron over here is collected by the electron acceptor. And then transferred along the electron transport chain. If you don't really know what we're talking about, this is the stages of this photosynthesis of the cycle, the light dependency. If we're talking about the light dependent chain, the electron transport chain over here, that's what we're talking about. So it's called and goes through the electron transport chain. And so that's what we're going to write here. So over here, I'll tell you where we're going to get the marks. Let me just finish it up. And then to produce 1 molecule of ATP. 1 molecule of ATP. And then we're going to mention how ATP is used. ATP is used in production. It's used in the production in GAP, off GAP, and GP. Okay, so over here, the marks that we're getting, first of all, is explaining. Oh, shoot, I forgot about photolysis. And then photolysis is. Okay, so the mark that we're getting over here is explaining what I don't get it. The mark we get is explaining what photolysis is displaying of water, and light is used in photolysis. Then we get one mark of saying that it reduced NADP to NADPH, the H plus ion, and that's one mark over here. And then the third mark over here would be to the excited electron, and then the parts of it going through the electron transport chain, and then making one molecule of ATP is one other point, and then used in production of GAP. Even though this is a stretch, but it is mentioned in the mark scheme, so I think we need to explain why we need ATP and how it refers back to photosynthesis. So that's an important part. Other things that is not mentioned here that we can talk about is that the electrons from photolysis, they're replaced because they're replaced by that are lost by chlorophyll to produce H plus ions. But over here, I don't think that makes sense with the chain of command, so I left it out. Otherwise, this just gives us 5 marks. One for here, over here, one here about the excited electron, one about the electron transport chain, one about ATP and production of GAP and GP. So that's a total of 5 marks. Then we have multiple choice. So we need to pick what are found in plants. Glycogen is not found in plants. Cellulose is, but that's going to be wrong, so we can cancel that out. Then we have cellulose and starch. That's the correct answer. If you look at the other ones, there's glycogen, which is actually not present in plants. It's only present in animals, so we need to talk about that. So make sure that you choose the correct one. Then describe how the carbon in these polysaccharides is returned to the atmosphere. This is a fairly simple question. It's 4 marks. So first of all, we talking about, it says how the carbon in these polysaccharides. So we need to take into account cellulose and starch. So we need to explain where cellulose and starch, where are they used. So over here, I'll say that cellulose is present in the cell wall, because the cellulose cell wall. So it's present in the cell wall, and when a plant dies, decomposers, we can explain decomposers like fungi and bacteria break down cellulose to simpler carbon compounds. Ignore my handwriting, it's really bad. To simpler carbon compounds. Okay, and we can explain those simpler carbon compounds. I will probably just leave it here. You can mention it if you want to. Obviously, cellulose is a large one, so we can say it breaks it down to glucose. We can say that it breaks it down to its monomers that it's made from, because there's a polysaccharides. They have it's made by units. So we can say whichever unit it's made from, which is probably going to be glucose or lactose or something else. We can also make up simpler compounds like they respire and it's released in the atmosphere as carbon dioxide. That will be there as well. But I'm just going to leave it here. Now I'm going to mention starch. Okay, so for starch is present in its present is as an energy store. I wouldn't say in, its presence as an energy store in plants. And made of glucose monomers. Made of glucose monomers. And so in respiration, it is broken down to get ATP for chemical reactions and CO2 is released. Into the atmosphere. If there's anything else I'd like to add, we can also talk about hydrolysis of glycosidic bonds in starch, but then again, I think it's a bit of a stretch here. These over here we've given where it's found in and how it respires, where it's found in and how it is given back to the atmosphere, so I think that will give us the 4 marks. If we wanted to talk about something else, we can talk about hydrolysis of glycosidic bonds in starch. There's also glycosidic bonds in cellulose. And we can also talk about which enzyme is going to be, so carbohydrates. And then it releases CO2 into the atmosphere. That works as well. It just really depends on what you're looking for. And that's it. We got 12 marks in this question. Then over here we've also done it in 12 minutes. So that counts for something as well. So we're going along with the time frame. Then we have to find the structure, describe the structure of a virus. Now over here, I feel like it's better if I make a text box because that's going to make it make more sense. So over here, let me just make us. So we all know the structure of a virus, or it should be quite easy at this point. So a virus, the things that we're going to talk about is it has a capsid. I can mention what a capsid is, which is a protein coat. Okay, then we can also say that it has nucleic acid, whether it be RNA or DNA. We can also talk about it contains some enzymes, like we have in HIV we have integrase and reverse transcriptase. No, we have integrase and reverse transcriptase. Yeah, I think that's the two that we have there. And genetic material in viruses can be double or single-stranded. Okay, so over here this is fairly simple. I give you like 5 points. We can choose any one of them. If I had to put them in order of importance, I'd say capsid is definitely the first one, then the nucleic acid one, and these two are more of like some of them have it, so we have to be careful about how we write it down. We can't just say that it contains all viruses contain enzymes or all viruses contains DNA, because that's not true. Some of them contains RNA. And yeah, so it just depends. So we need to be very careful with how we word it. And then we can't say that it can be only double-stranded because that's not going to gain us marks. So we can just choose and mix and match. Explain why there's a delay following disinfection before the number of virus particles increases. Okay, let me read it again because I didn't read the question. Delay in. Okay, so it says the human body responds to a virus infection by producing interferons and antibodies. So first of all, just for my own stuff, I'll put in the words over here. So interferon and antibodies. What are interferons? They make sure they're chemicals which make sure that other cells don't get infected by the virus. So now that I know about that, I can, if it's possible, you can just write it down here. And then antibodies, we know how they're made. That's the humoral and cell-mediated response, all of those, the B effector cells, and how they turn into plasma cells, and then they give antibodies, and how they affect it. And then the graph shows the change in the number of virus particles, the level of interferon, and the level of antibodies in a person a week following the infection. So this is the number of virus particles. And you can see that it increases and then it decreases. So we have to say that why is there a delay before it increases? Like over here, why is there a delay? So that's what we're supposed to make. So obviously for this is a 2 mark questions, we have to give 2 points. So that it takes time for the synthesis. So we can just start off with that the virus is multiplying. So the virus is multiplying, and it takes time for assembling of a virus using host cell materials. And this is going to be our first point. And the second point would be that as they reproduce, it takes time for synthesis of DNA. It takes time for synthesis of DNA. Again, I can't say DNA and RNA. I have to be careful that I say or. The wording really matters, and that's basically it. So one point was for saying that it takes time for assembling a virus. If I were to say, I actually would have given the second point first, this point first, before going over to the assembling of a virus. You can like inverse it if it makes more sense for that way. Multiplying, I don't think it is true, but I don't think it will give you the mark because it's very vague. We need to explain how, why would it take time for it to multiply? What is it that it's doing that takes time? Now we need to explain the change in number of viruses from day 1 to day 5. So over here, let's have a look here. Okay. Wait a second. Yeah, I think that's perfect. And so over here, if you look at the graph, this is infection. This is day 7. So of day 5 will probably be somewhere around here. Definitely. So day 1 to day 5. So it's increasing. Virus is increasing over here. So we need to explain the change in the number of viruses. So then say describe it. So we're not going to say that it's increasing. We're going to just go straight to the point and explain why is it changing. So for here, I will write that more host cells are getting destroyed because the viruses, they're attacking host cells, they're multiplying, and they're using its material. Whatever they have, the cell wall, they're just using it. The cell bursts, it gets destroyed, and so more cells, host cells are getting destroyed. So then more, actually I'll give it as another point. So more viral particles are released, and so more cells can be affected. So it's like a chain, because viruses, that's the reason that they're so deadly, because they can attach to it, it multiplies and increases the number, and now it can attach more, so it's like exponentially increasing. And that's where the third question links to it. Describe the role of interferons. So as I mentioned before, I actually did give you the definition of what interferons do. They inhibit viral replication. We can say stops viral replication, because they make sure that they can't attach anymore to more host cells, so that other cells cannot be infected. So that's exactly what we're going to say here. And I'm not going to say they're chemicals, because it's not telling us to tell what they are. We have to say what they do. So interferons inhibit viral replication. That's one point. And then we're going to say that it prevents attachments to host cells, so other cells cannot get infected. Okay. Explain why there is a delay before the levels of antibodies start to rise. So first, it's important to note that this is a primary infection. Just by looking at the graph, you can tell that it's a primary infection. It's the first time this virus has entered the body. So antibodies, they need to be formed first. Okay, so I'm going to start off saying that this is a primary infection. So antibodies have to be made. Then we're going to explain how antibodies are made. So we're going to start about T killer, T memory cells, B cells, B effector cells, how they differentiate to plasma cells and release antibodies. We have to mention that it's the humoral response. So a humoral response takes place. I can definitely write it better, but it proves the point. It's not the best language to use, but I think it gives us the mark. A humoral response takes place, and then we have to explain it. So the macrophage engulfs the pathogen. And presents its antigen, antigen APC, and then and presents its antigen as a APC, which is an antigen presenting cell. Everyone should know that. And then it binds to T helper cells, and then it activates to form T killer and T memory cells. And then the T cell, it binds to a complementary B cell, which releases cytokines and makes B effector and B memory cells. And then B effector differentiates to plasma cells, which release antibodies. That's the whole thing. I'm just going to write that down right now. So then it binds to a T helper cell. The T helper cell and activates. Actually, was just activated and activates to form T killer. Oh god, I forgot. Actually, activates to form T killer and T memory cells. Then the T cell binds to complementary. The language in biology is very important, so you have to say words like inhibit, complementary, antigens. We have to say all of these words to get the mark, so because it just matters that much in biology. So binds to a complementary B cell and release cytokines and makes B effector and B memory cells. And then the B effector differentiate to, did I write that wrong? Differentiate, oh there's a single T, differentiate to plasma cell which releases antibodies. Okay, over here I'm just gonna turn it to science. And that's basically it. That's our 4-mark question. There we are. So we should have spent about like 25 minutes on this. I spent 26, but I was talking, so hopefully it works out well. Then over here we're talking about cyanobacteria. It's Lake Vesidorve in Finland that became polluted a few years ago. This resulted in the lake becoming overpopulated with cyanobacteria. Large numbers of cyanobacteria make the water very green and cloudy. Cyanobacteria are synthetic prokaryotic organisms. Cyanobacteria are classified in the domain bacteria. Name one other domain. This is a simple question, you don't even need like the question to make it make sense. It could be anything. It can be in eukaryota, it could be archaebacteria, it could be, yeah, those are the 2 ones, but archaeota is what I'm gonna say. Eukaryota. I'm pretty sure I wrote that right. Why is it coming off as wrong? No, it's supposed to be top. There. Then over here we have to say that organisms can be classified as belonging to this is AS to be fair, but we still need to know it, so organisms belonging to domains using, that's called molecular phylogeny, so that's the answer. And over here, place a cross next to the structures found in cyanobacteria. So it says it's photosynthetic, but we don't really know if that chloroplasts or not. It's not a plant, so it's going to be hard for us to see if it's going to be chlorophyll in it. It could be some organisms, they don't need that to photosynthesize. They're prokaryotic, so that means that they have the 70S ribosomes, and then it's a bacteria. So if we know like the bacteria structure right here, this is awful, but we're gonna go with it. That's like an example. That's the loop of DNA. They have a circular DNA, and then they have a plasmid. Some of them have a plasmid. There's a flagella, there's pili over here, which helps, and then they have like these small tiny vacuole thingies as well. That's basically a kind of creepy drawing of a bacterium. But yeah, that's basically it. So it has a loop of DNA, and it has small 70S ribosomes, which is smaller, and they have large. Even if they didn't give you large and small, we should know that they have 70S, because it's a bacterium, so we have to follow that. It won't be chlorophyll because we don't exactly know what it uses to synthesize. It's going to be quite a long shot to assume that it has chloroplasts. Suggest why it could be green cloudy water could be a problem for organisms in Lake Vesidorve. Okay, so I would say that there will be because organisms, okay, first of all, plants, let's say plants, okay, let me just write that down. So my chain of reasoning would be that because it's green cloudy water, plants that are living inside the lake, they need light to photosynthesize, and because it's green and cloudy, there will be less light for photosynthesis, and that means that it's going to hinder their growth. And since there's fewer plants, that would mean that there's less food for other organisms as well. because they're all related, it's an ecosystem, and that's why it's going to be a problem for organisms overall in Lake Visijorvi. So let's say, so green cloudy water means less light for photosynthesis, and so will hinder growth of plants. Less, how did I write photosynthesis wrong? Okay, I didn't add an O. Less plants would affect other organisms that prey on those plants, and so less food for them, as they'll decrease in numbers. Thus they will decrease in, decrease in number. There we go. Okay, then scientists can alter the abundance of organisms in a habitat by removing or introducing organisms. This is called biomanipulation. The effects of pollution can be reversed by removing 80% of the roach from the lake. Other fish that eat roach were introduced into Lake Visijorvi. Roach are fish that eat zooplankton. Zooplankton eats cyanobacterium. As a result of this biomanipulation, the water in Lake Visijorvi became clear. Explain why the water became clear. Okay, so they're doing, what they're trying to do over here is that they are decreasing the number of the roach. They're removing them. 80% of them are being removed, so that the number of zooplanktons would increase. If the number of zooplanktons would increase, that means that they would eat more cyanobacteria, and the number of cyanobacteria would decrease. And because there's going to be less cyanobacteria, is the reason why there's green cloudy water. So if that's going to decrease, that means the water will be clear. So that's why we're gonna say that. So we're gonna say less roaches mean fewer zooplankton. Zooplankton will be eaten. This is a very simple question for a 4 marker, so you should definitely be able to get the mark. Less zooplanktons will be eaten. So then zooplankton numbers will increase. Okay. More zooplankton will eat cyanobacteria. So cyanobacteria would decrease in number, and then water will become clear. This was a very straightforward question. Clearer. And that's our 4 marks. The last part is just for me. You shouldn't, like, it's not going to give you a point, it's just going to make you, flow naturally. So that's basically it, and we're going to start with question 4 later in another video. This is going to be the end of here. And yeah, that's what we did right now. I think writing the questions over here is way better. I should use a text box more often because it just makes it so much more simpler here. And I think you should ignore the drawing. It doesn't really make sense unless I tell you what it's about. And it wasn't that hard for now. I think the first 2 questions were very simple. And if you have like the knowledge for it, you'll be able to do it. That's it. And then we're going to start from question 4 later.